Hello everyone, and thank you for joining us for this Room Now Giant Cell Arteritis panel from ACR 2020. I'm Richard Conway from Dublin, Ireland, and I'm joined by a truly outstanding global group of giant cell arteritis experts to discuss some abstracts from ACR 2020. We have Sarah Mackie from Leeds in the United Kingdom, Len Calabrese from Cleveland in the United States, and David Liew from Melbourne in Australia. So the first abstract we're going to discuss is the one on maverilumab in giant cell arteritis. So Sarah, I might ask you to start off on this one.
So this was a report that was presented earlier today by Maria Sid. So it's a phase 2 randomized placebo-controlled trial of maverilumab versus placebo in addition to a prednisolone 26-week taper. So eligible patients had active giant cell arteritis and they were either new onset or they were relapsing refractory. And 70 patients were recruited. The primary outcome was time to first flare, and the key secondary outcome was sustained remission to 26 weeks. The headline results of the study is that it met its primary and its key secondary endpoint with a hazard ratio of 0.38 for time to flare, and 83% of the patients in the treatment intervention arm achieved sustained remission compared to 50% in the placebo arm. So it looks exciting. It's a phase 2 study. We don't have the full study report, so there's still a lot of things that we'd love to know about the results of the trial. But it's very exciting because it's an exciting result on a new biologic, which is not so far licensed for any indication as yet, to my knowledge.
Len, I wonder if you might tell us a little bit about how maverilumab works and why it might be an attractive option in terms of GCA pathogenesis.
Yeah, well, I volunteered to do that, and I was thinking here, GM-CSF is such an interesting molecule. It's in a short generation ago was thought to be a hemopoietic growth factor, and now we recognize it as a central mediator in inflammatory processes, mediation of pain. It's in very advanced trials in a number of diseases, particularly rheumatoid arthritis. And it is not totally clear in terms of a crisp immunologic story, which pathways are being involved. It is obviously produced by hemopoietic cells and viscerosomatic cells that can signal hemopoietic cells and viscerosomatic cells. So it's very central, kind of like that IL-6 centrality. Recent studies in the Lancet for another molecule, otilimab, recently in RA, a big biomarker study shows that very little moves outside of acute phase reactants and a lot of things that we're looking for. So preclinical modeling suggests that it is active at the endothelial interface, and these are pretty impressive data, and it's all going to be about risks and benefits, and the safety signal that was released there was quite favorable at this juncture. There's obviously things that we're concerned about from preclinical modeling based on things like pulmonary alveolar proteinosis, and that really has not been seen in any of the global programs thus far. Infections will be on the lookout, leukopenia will be on the lookout, obviously. And then, there's some modeling in bowel and even things like autoimmune thyroid disease that could be looked at. But in this little short trial on the basis of the publication, it's looking pretty good so far.
Was maverilumab before this? It was studied quite a bit in rheumatoid arthritis, wasn't it?
Yes. Yes, and it is still advancing in the space, and it is effective. The question is, that on a lot of people's minds is there room for another anti-cytokine in this very, very crowded space? But there are advanced clinical trials going on with another molecule, otilimab, which targets the cytokine instead of the receptor, and we'll see.
Great. So, David, GCA is definitely not a space where there's a lot of cytokine therapies available. Where do you think this might fit into our treatment armamentarium?
Well, I think we're yet to see. Obviously, this is phase 2 data. This is early. We haven't seen this fully reported yet at this meeting. It's late breaking abstract. So I think there's a lot to play out with this. I mean, the idea is enticing. And from my simple point of view, really, in the Conway paradigm of Th1 and Th17, that it might be able to affect both pathways plausibly, that might be impactful. We've seen, I'm biased because GMCSF, with a lot of the developmental work was done from Melbourne, Australia. So I'm a bit biased on that point of view. And I think there are some institutions here which will get some royalties if it comes through. So I don't have any stake in them. But, we've seen really nice translational data. So Maria said actually presented some stuff at ULI 2019 showing the genetic signature at the temporal artery. So I think there's been a nice story to this. But where's it going to sit? Well, I think, we've got to see what happens in the short term, in that early phase in giant cell arteritis when it comes through. We won't know that now. Obviously, small numbers. We still need to see the full reports on this. But secondly, what I'm really interested in is potentially what it looks like over a longer course of time. Really, of course, what's the steroid bearing potential, but then maybe it has plausibly greater effects on some of the actual intravascular changes that we might see with GCA. Now that's highly speculative, and I guess I'm in a position where I can speculate about these things as a neutral observer. But yeah, I think there's potential that it might have. It might fill a new space, or it may well be in competition with tocilizumab and other IL-6 inhibiting monoclonal antibodies.
Yeah, so certainly I think that dual targeting of the T helper cell pathways, it is enticing in terms of a longer term treatment, a more complete disease control, and maybe we could completely get GCA into remission rather than just dampening it down as we fear might be happening with some of our other treatments. So I think we can all agree it's a really exciting therapy at the moment. It's in very early stages of development, but hopefully more evidence will be coming out as we go through the next year or so on this agent. The second abstract that we felt was very interesting from this study was from Peter Villager and his group in Bern. It was the Gusto study, combining an ultra-short steroid course with tocilizumab. And David, I might ask you to tell us a little bit about that study.
Absolutely. Well, I think that overall in GCA, the paradigm's always been about how much steroid can we get rid of. And that we've lived with a long time with giving people a lot of steroids, seeing people gain weight in front of our eyes and get all the complications that we associate with high-dose corticosteroid use. And then tocilizumab changed all of that. And we've seen the quite marked steroid-sparing capacity from that. But the question always is, can we go further? I guess in a 26-week wean or 52-week wean of steroid, there's still quite a lot of steroid going on, more than we'd consider giving to something, to patients with diseases like rheumatoid arthritis. So what this was a investigator-initiated a small study, open label clinical trial from Peter Villinger's group, the group which reported the first clinical data in GCA with tocilizumab. And what they did was they had 3 days of pulse IV methylprednisolone, 500 milligrams, and then no more steroid after that, tocilizumab after that, and then seeing what happened beyond that. And I remember a few years ago at this meeting, there had been a report from Japan where they didn't use any steroid and used tocilizumab. And I think there's a lot of faith at that point because we know that tocilizumab doesn't work as quickly as corticosteroids. And that's why G-Actors had a steroid wean built in to the tocilizumab as well. So just to give the high level the high-level outcomes. There were 18 patients who got through to the complete analysis. And of those, 14 of those achieved remission, but it took them a mean of 11 weeks. 13 of them were relapse-free at over the 24 weeks. But then there were 3 patients who were non-responders, one of whom only had PMI symptoms, but 2 had cranial symptoms, one of whom had a new onset AION and had permanent visual loss. So that seems like that's a pretty big thing there. And also to note that the inflammatory markers took some time to settle, really. I mean, even with tocilizumab, not entirely sure exactly when, but somewhere between 10 days and 4 weeks in the reporting, we know that that took that long for the inflammatory markers to normalize. Is that kind of time period something that's acceptable or not?
So yeah, and Len, I might ask you, in terms of the immunopathological mechanism or plausibility of a strategy of giving this very short, very high dose steroids in combination with IL-6 inhibition, is that something that makes logical sense to do?
You know, I don't want to be hypercritical. I mean, this is a very important study, and these guys are very smart, and they, designed it in a very acute way. I would not have designed that study, and I don't really think that I would have equipoise to even put a patient in that study, to be honest with you. And I'll say that for a couple of reasons, and this is all hand waving, and if I had a glass of wine, I'd be waving that around too. One, I don't think steroids, pulse steroids have ever cured GCA. There's this copious literature on pulse steroids for people presenting with, acute visual loss, where, these crazy ophthalmologists give them 1 gram or 2 grams a day for 3 or 4 or 5 days, et cetera. And still they have difficulty of getting people off of steroids. So, I mean, you have that in the background. Secondly, we have known from practice that the greatest risk to permanent visual loss is a history of previous visual loss. Those are our most vulnerable patients. And we know that if we can treat them and get them through the first 2 to 3 weeks, visual loss after that is extraordinarily uncommon. So I would have opted for giving them steroids for maybe 3 weeks. We went from thinking that, 6 months is short steroids. I mean, we haven't really been doing that either yet. 3 days is, I think it's just a bridge too far right now. But I don't say that in any other way other than Monday morning looking back at this right now, but I wouldn't have done it that way.
So, Sarah, what do you think about where we're going with steroids in giant cell arteritis and where we might end up?
This makes me wonder about whether maybe we'll always need a little bit of steroid together with biologic therapies. So there was one patient in the treatment arm of Jiacta who had acute ischemic optic neuropathy who recovered with increase of steroid, but they did have AION, and they had they were on the biologic arm. There was the patient in Seresta who which had a 3-month taper, who had visual loss, and again that was in the treatment arm, and it's just very short steroid tapers, and you wonder whether the systemic effects of the cytokine inhibition are blunting some of those early warning signals of GCA relapse. And so, or maybe the trials have just been unlucky, and maybe we always have some visual loss in our cohorts and we just choose to ignore it. So I don't know. It's just individual cases, and you can't make huge decisions for the world about guideline decisions based on like n=1 per trial. But it does make me wonder whether a little bit at a tiny dose of steroids maybe might protect some of our patients, maybe our oldest patients, the ones that have got highest risk of visual loss. I don't know. There's no one. I don't think we can say from the evidence. But I get a little bit nervous with patients who have those highest risk patients that Len was referring to, the oldest ones, the ones who have previously had transient visual loss. Just worry a little bit about them taking them completely off steroids. if they're also on a cytokine inhibitor as well.
Yeah,
You're
I
not saying
agree.
we're turning, sorry. Oh no,
Okay.
you're not saying we're turning a blind eye to our fallibility in this disease, are you?
I think there's a lot of wishful thinking in giant cell arteritis, isn't there? And see what we want to see. So yeah.
I think that is probably a nice note to end this conversation on.
Very nice.
And I would like to thank everyone for watching, and remember to go to Room now for more updates from ACR 2020.
Very much.
Thanks.